Hello everyone! Today, my team and I are going to present our infographic. Just for the triangle, let's start! Signaling pathways in embryonic development. Embryonic development is characterized by the careful regulation of the cellular behavior so that cells proliferate, migrate, differentiate, and form tissues. These processes are genetically controlled and depend on the cells and signaling pathways that coordinate the cellular interaction, leading to organogenesis. Signaling pathways operate during development, acting as cells develop. In recent years, many advances have been made in identifying the signals that act during embryo development and in understanding their properties and function, which is also important for pathology and human evolution. In addition, many of these mechanisms are involved in adult tissues homeostasis and adult tissues regeneration. Understanding signaling pathways is important for several reasons. Number one, because gives information of mechanisms of the cell's function and interaction for morphogenesis and organogenesis. Number two, it allows to discover the basic of congenital malformation. And number three, it allows the identification of possible causes of diseases in the adult organism.
Among the signaling pathways with the roles in development is the WNT pathway. WNTs can bind to multiple receptor complexes and determine signals. For example, WNT primary, canonical WNT, beta-catenin dependent, non-canonical WNT, and WNT catenin. An important fact is that the signaling signal can elicit diverse cellular responses depending on the cell type, context, and development. And now we have some cellular responses, such as canonical WNT. The role of canonical WNT signaling in eye development is in the development in patterning of the ocular tissue, differentiation of retinal pigment epithelium, and morphogenesis in the optic cortex. And importantly, fact misregulation of the signaling cascade can lead to eye malformation and disease. Another important signaling pathway is Notch, a cross-membrane protein. Notch has many functions in organ formation and adult homeostasis, including cell determination and stem cell maintenance. Also an important role in somatogenesis and the role of FBXW7 protein in regulating the turnover of Notch intracell domain in development and cancer.
Crosstalk is a mechanism that allows cells to respond differently to same signal, producing the diverse cellular behaviors that are needed with tissues and organs. The FGF signaling is important in the developing spinal cord. The nervous system arises from neural of an early neuromesodermal located at the caudal part of the embryo. The FGF signaling could be related to ability to promote regeneration in the injured spinal cord of the adults. The Notch signaling and BLHL transcription factors opening avenues for possible therapeutic approaches in hearing impairment present a crucial role of autophagy regulated process of degradation and recycling of cellular components, invertebrate inner ear formation. The SHH is important pathway for congenital malformation in humans and for the generation of limb morphological diversity during evolution. The SOX9 in the expression patterns among species that have specific morphologies.
The integration of extrinsic and intrinsic regulatory signals is essential for the organ formation, as well as in the formation of the epicardium. This is the outermost layer of the heart that not only serves as the content covering of this organ, but also appears to play a role in regeneration. Therefore, understanding the basis of its development can have important therapeutic implications. Importantly, it also appears to be involved in the establishment and fusion of satellite cells. The stems are resident in adult muscles, thus opening up new avenues for the development of regenerative therapies. Apart from the intrinsic molecular signals, the interaction of the muscle cell with the surrounding tissues, bone, cartilage, tendon, and ligament is essential for the correct assembly of the musculoskeletal system during development and to maintain adult homeostasis. An emerging topic in developmental biology is the control of tissue morphogenesis by psychical forces, mechanotransduction. It reminds us that cells from embryos and adult organisms are not present in isolation, but are embedded in extracellular matrices in complex tissues. Cells adhere to the ECM and detect its mechanical properties. Normally, in vitro experimental conditions do not fully reproduce this environment, which is nonetheless critical for physiological cellular response to